Hello everyone, my name is Fayaz Khan Sahil and today we're gonna learn about human digestion. This topic gonna be really interesting. Think about any fruit or any food you eat. Suppose this is a apple, okay? It doesn't look like an apple, but it might look like an apple. So suppose this is an apple, right? Apple is a large molecules, right? It cannot be absorbed by the wall of our intestine, right? So these larger molecules needed to be broken down into smaller molecule so that this can be absorbed by the small intestine, then transfer into the bloodstream to the cells where they require energy, right? So, the process of breaking down food into smaller absorbable molecule is known as digestion. There are two forms of digestion, physical digestion and chemical digestion. Sorry. Physical digestion involves breaking down the food into smaller pieces without chemically altering its structure. It is mainly done by the teeth, and the stomach helps to digest the food further. And let's move on to another type of digestion, which is chemical digestion. Chemical digestion uses enzyme to break down the larger molecules into smaller soluble one through chemical reactions, right? Now let's move on to the other part of our digestion, which is teeth. Teeth play an important role in both taking in food and breaking down it physically, right? They bite off the chunk of food and then crush or grind into smaller pieces. This increases the surface area for the food, making it easier for the digestive enzyme to act on it. How come it increases the surface area of the food? Suppose this is a food, okay? This is a food. As you can see, if you break down this food into smaller chunks, it actually increases the surface area so that enzyme can work on it. It also allows the food to be soluble substances so that it dissolves more easily in the saliva, right? And before that, what does saliva contain? Saliva contain an enzyme known as amylase, right? Amylase, which break down starch, by the way. Now let's look at the structure of the teeth. The structure of the teeth shown in figure 7.10. Each teeth is set into the gum, and the part visible above the gum is coated as enamel. This is the enamel. Enamel is a very tough substance that covers the outer surface of the teeth, right? If you touch your teeth, you can see the surface that is hard. It is enamel actually. It is very tough substance, as I mentioned. It is also interesting fact. It is one of the hardest material produced by animal, and it is very resistant to damage. However, it can be worn by the bacteria. So what does bacteria release? These bacteria release acids which actually can digest this enamel, which actually can dissolve this enamel, causing tooth decay. Now let's move on to the more part of the teeth. The teeth also called dentine. Dentine, as you can see, this is a structure is dentine. Dentine is a living tissue that lies. The enamel is non-living, okay? It isn't living, but dentine are living tissue that which is rather like bone. Dentine is quite hard, but not as hard as enamel, right? It has channel into which it contains living cytoplasm. In the middle of the teeth, there is a nervous system and there is blood. That's why, because of this nerve, you can feel the pain in your teeth. Without nerve, you cannot feel it. The part of the teeth that is embedded, okay, that is embedded in the gum, covered with a material that hold the teeth into the gum, okay, we call it cements, right? Cements hold the teeth with the gum. This allow it to move slightly when biting. Again, sorry, I'm a little bit sick. Please stay with me, and I'm really new to the YouTube thing, so forgive me for any mistakes, because I'm a bit nervous to teach you guys. So let's see. I'm very introverted as a person, so it's hard for me to come out and just teach in online where I don't know where you guys are from and how you guys are judging me. Don't judge me for at least 1 year. Don't judge me. I'm trying my best. Okay. Now we will look at several. There are 4 types of teeth that are found in mammals, particularly in human. Incisor. As you can see, this is a front view. This is a side view. Incisor are sharp, chisel-shaped teeth located at the front of your mouth. These are the teeth that is located at the front of your mouth. They are used to bite off a piece of food. For example, if you eat an apple, the teeth you bite off with is actually incisor. Then we have canines. Canines are pointed, right? These are very pointed. Their function are similar to the incisor but more prominent in carnivore, where they help to grip and kill prey, right? In tigers, these canines are very, very important in tiger. And we have premolar, right? And interestingly, your sir has a cavity in both premolar and molar teeth, and this is very painful. So the premolar are found between the canines and the molars. The teeth have broad, as you can see, it is broader than these teeth, right? The teeth are broad, rich surface, and are used for grinding food into smaller pieces. So these are the teeth are actually, the premolar and molar are used for physical digestion. They are actually used for physical digestion. Sorry for my handwriting too. Molar are larger than premolar, okay? Molar are much larger than premolar and have a broad, rigid surface located at the back of your mouth. They are mainly used for chewing and grinding food. In human, the last molar are at the very back, called the wisdom teeth, as you all know, which is usually appear later in life. Now let's look at chemical digestion. This is very interesting. Chemical digestion are actually occur because of the enzymes, okay? For example, in stomach we have various protease enzymes, for example pepsin, trypsin, that break down proteins, right? So these digestive enzyme to reach the food molecule more effectively and begin the breakdown. For example, amylase found in the salivary gland in the mouth, where it acts in the mouth. Amylase breakdown starch, right? Amylase breakdown starch. Protease, on the other hand, found by the wall of the stomach by the pancreas. It is released by the both stomach and the both pancreases. Their main function is basically to break down protein into amino acids. Okay, lipase. Lipase are actually secreted by the pancreases, which break down fat. Now let's look at human digestive system. Human digestive system, okay, the stomach. We are particularly talking about the stomach. The enzymes are influenced by both temperature and pH. The enzyme involved in a digestion in the human body work best at 38 degrees Celsius. So your question might be, what will happen if your body temperature started to increase? Look, enzymes are protein, okay? In higher temperature, this protein might denature. Okay? However, there are different digestive enzymes have different optimum pH level. For example, the enzyme found in the stomach pH is pH 2 actually. So these protease enzyme work best in this pH, and also there are hydrochloric acids in the stomach that actually kill the bacteria. Now, in contrast, the enzyme that function in duodenum needs slightly alkaline environment above pH 7 to work efficiently. This is achieved by the bile and the pancreatic juice. which contain alkaline substances that neutralize the acid content coming from the stomach. Now let's move on to the function of the enzymes. As you can see, the amylase in the mouth actually break down the starch into sugar called maltose. Okay, what is maltose? Maltose is very simple and beautiful. When two glucose molecules join together, it is called maltose. Okay, if you chew starchy food for a while, you might notice a sweet taste. This is because the amylase in your saliva started to break down the starch into maltose. Now let's look at each maltose molecule is made up of two glucose unit, as I have explained. Although maltose is smaller than starch, but it is still larger to be absorbed by the body, right? So the enzyme maltase break down the maltose into individual glucose. Maltase is produced by the cell that line the small intestine called epithelium, a tissue that covers the surface inside the body, such as the wall of the digestive tract. Unlike some enzyme, maltase doesn't enter the lumen of the small intestine. Instead, it stays attached to the membrane of the epithelial cell, meaning maltose is broken down right on the surface of this cell. Now we have other proteases enzymes such as pepsin and trypsin. Okay, the protease in the stomach is called pepsin, which work best in acidic pH, which is about 2. It is released in a fluid called gastric juice, which is secreted by the stomach lining and contain both pepsin and hydrochloric acid. The acid not only creates the ideal environment for pepsin to function, it also helps to kill harmful microbes in the body. Okay, so this pepsin enzyme require a pH of 2 for function efficiently, and also they work best in hydrochloric acid. Okay, the acid actually keep the structure of a pepsin in the right direction. Now we have what happened in the duodenum. Another protease called trypsin is produced by the pancreas. Unlike pepsin, trypsin work best slightly in alkaline condition. Oh, I'm really sorry. The trypsin is actually found in the pancreas. I misunderstood it. I actually mistakenly said that. The trypsin work best in slightly alkaline condition with an optimum pH of above 7. This is why acidic mixture from the stomach must be neutralized by the alkaline substance. Look, the stomach, for example, this is a stomach. It has acids, right? If it flow to the small intestine, it might cause harm to the cell, right? So the acid need to be neutralized. So the duodenum both pancreas and duodenum releases alkaline substances that actually neutralizes the acidic environment. Both pepsin and trypsin break down protein into amino acid, which we just explained, which are small enough to be absorbed into the bloodstream. Now let's look at fat digestion. Okay, fat and oil do not dissolve in water, so they enter the digestive system and they remain as separate droplets. So in the blood system they will remain as droplet. So these are like fat droplets. These are like fat droplets, right? This fat need to be broken down. Bile, which is secreted from the liver, actually break down the larger. Suppose this is a fat molecule, the bile will break down it smaller molecule, right? That is a particular function of digestion, right? These tiny droplets are spread out throughout the watery fluid, forming a mixture known as emulsion, while small droplets of one liquid are evenly dispersed in another. This process is known as emulsification. It increases the surface area of the fat, making it easier for the enzyme to digest it. Okay, we are up to this for today, as I can say. Thank you if you're watching this video, and please do subscribe to my channel. I'm trying my best. I know I'm a bit shaky because I'm really nervous to talk in front of the in front of all of you guys. So please stay with me. Bye bye. See you guys in the next class.